Hello, everybody. My name is Peyton Harrison. I am an undergraduate cell and molecular biology student completing research at North Dakota State University and part of the CIDR REU program. Today, I'm going to walk you through one of the most essential concepts that I rely on daily within this program, genetics and the central dogma. Together, we're going to explore the remarkable code of life, uncovering how the body reads, processes, copies, and expresses genetic information. One question that I would like for you all to ponder first is, what do you think of when I say the word genetics? I asked this very question to my fellow REU students, and here's what they had to say.
So, yeah, not a lot. Like the main words that are coming to mind are like DNA. Okay. I remember like those little gummy bear diagrams that I thought were cute. It's like where it's like half a gummy bear, half a gummy bear. So yeah, when I think of genetics, I just think of like, yeah, like what what I look like and why I look the way I do versus like my sibling.
Inheriting traits from your either from your parents or like further down the line, doing like genetic testing and seeing if you have like some disease that's like in your family, which contribute to building you into the person that you are biologically.
Of DNA and like genomes, I guess. Things that, affect what we look like and like our eye color and stuff.
So, are they right? Yeah, all of these students are correct in their own ways. From gummy bear diagrams to DNA strands, eye colors, and the differences between siblings, these are all often associated with genetics. But what exactly is it that ties them all together? The answer lies in the central dogma. The central dogma describes the flow of genetic information from DNA to RNA to proteins or polypeptides through two processes called transcription and translation. It's through the central dogma that we begin to understand how the information from within our genes becomes something that we can actually see, or what's known as our phenotype. As we can see in the diagram, the phenotype of the very first generation of gummy bears is red and white. As the generations continue, the phenotypes change drastically depending on their parent's phenotype. Let's break this process down from the beginning. DNA. DNA is frequently projected as this double helix. It is located within the cell's nucleus in these structures called chromosomes. DNA is like a master instruction manual for building and operating living organisms. DNA is made using four chemical bases: adenine, cytosine, thymine, and guanine. Adenine pairs with thymine, and cytosine pairs with guanine to form the famous double helix structure. Keep this complementary base pairing in mind. It's going to come into play soon. Each gene is a segment of DNA that codes for a specific protein. The genetic information that is stored within the DNA flows into a new form, RNA. RNA is a single-stranded photocopy of DNA, but instead of thymine, RNA uses uracil. RNA is the messenger that carries the DNA's code into the ribosome. It functions as the delivery service of the system. This process of changing the DNA into RNA is known as transcription. Following transcription is a process called translation. Its job is to make proteins. Oh, no, not those types of proteins. I mean these types of proteins. They're a molecule made of various polypeptides that are folded into a specific 3D structure. The process of translation builds proteins, turning RNA into a functional protein. But how exactly is this done? RNA is read 3 bases at a time. This is also known as a codon. Using this table, we can see that each codon corresponds to one amino acid. Link the various amino acids together, fold it into a 3D shape, and boom, you've got yourself a protein. So now what? What exactly is it that proteins can do? Proteins are the absolute MVPs of the body, in my opinion. They function as channels, help with structural integrity, they can act as enzymes and antibodies, they send signals, and much, much more. It is all dependent on the proteins made from your personal genetic code. Proteins can perform cell functions that result in gene expression. Frequently used examples of expressible genes in middle and high schools include the presence of a widow's peak hairline, your hair type, the ability to roll your tongue, if you have freckles or not, and earlobe types. However, genes also contribute to other phenotypes, including serious conditions such as inheritable diseases. Considering that our genes are so powerful, there have been various myths over time that oversimplify just how complex our genetics really are. Given the role that genes play, there is no surprise that the misuse and oversimplification of genetics is, unfortunately, commonly seen throughout social media. It is easy to begin Googling what is a myth or what is fact regarding common statements about such a complex system. Popular media often promotes misleading ideas about how genes work, and believing these myths can be both misleading and harmful. So in these last few minutes, we are going to be addressing what some of these common myths are and what they imply if they are misrepresented. Without further ado, let's bust some of these myths. Number 1. Genes determine everything about you. This concept is known as genetic determinism, the idea that our DNA is our destiny, therefore our traits are inevitable. This is untrue, because genes are part of a system that is incredibly complex, including factors from your development, given to you by your biological parents, or other factors such as your environment and lifestyle choices. What I'm saying is that human traits are incredibly complex. Therefore, this statement is an oversimplification for just how intricate our genes are, and how other factors need to be considered when discussing the nature of many traits. Number 2, genetic test results can tell you everything. While yes, genetic tests often provide ancestry information and good risk assessment, they're also not 100% certain, especially when it comes to complex diseases. Therefore, misinterpreting your results can lead to either false reassurance or unnecessary anxieties. This is why professionals emphasize that the interpretation of your results requires context, family history, and follow-up testing. Number 3, there is a single gene for complex traits. One of the examples that I found bouncing around the internet is that it is believed that there is a single gene that can determine either your intelligence or your sexual orientation. Not that either of these is mutually exclusive. Traits like these are what's known as polygenic, meaning that they are influenced by many other genes. There are these big complex webs that are unique to every individual based on their ancestry. Other factors that may impact these traits include your upbringing, culture, any birth complications, your education, immunity, trauma, training, gender roles, and much, much more. The way that we talk about genes can shape the way that people are treated. Trying to reduce a human identity to 1 or 2 gene variants is both scientifically false and socially dangerous. Today, there are many people who use genetics as an excuse to force hatred on certain groups of people. This is why the language of researchers use today advocates for an intersectional approach to avoid this genetic discrimination. If you are curious to learn more about genetics, I highly recommend the NIH or NHGRI websites. They offer a plethora of information on genetics and genomics. Now that you have your understanding of the central dogma, I invite you all to explore the current research projects that are driving both scientific and medical breakthroughs. That is your overview on the complexities of modern-day genetics and the central dogma. If you are open to learning more about your health, your identity, or how science shapes society, understanding this concept is a fantastic first step. Thank you all so much for watching, and I invite you all to stay curious and stay kind.